Hello and welcome to my channel Biology with Priya. Today the topic for this video is structure of seed part 2. Topics to be covered in this video are dicot and monocot seeds, structure of bean seed, structure of maize seed and some important questions. Now to take a review of the previous video of seed, a plant begins its life with a seed and the seed develops from a mature ovule after fertilization and is enclosed within the fruit. A seed contains two main parts, seed coat and embryo. Monocot and dicot seeds. Based on the number of cotyledons, seeds are either dicot or monocot seeds. Dicot seeds contain two cotyledons, for example, pea, bean, and monocot seeds contain only one cotyledon, for example, in maize, grain, maize, rice, and wheat. Now, based on the presence or absence of endosperm, they are classified as albuminous and exalbuminous. Now, these albuminous seeds are also called as endospermic seeds. They have thin and papery cotyledons and the food is stored in the endosperm. For example, in poppy and castor seeds. Exalbuminous seeds or non-endospermic seeds have fleshy cotyledons as they store food. For example, grass. Structure of bean seed. It is a dicot seed as it contains two cotyledons. Bean seed is kidney shaped and is covered by a hard seed coat. Seed coat consists of two layers, testa and tegmen. Testa is the outer hard brownish covering and tegmen, which is the inner thin and pale yellow covering. Seed coat protects the inner part of the seed from injury and infection. Hilum is a whitish scare on the concave side of the seed. It is the point where ovule is attached to the ovary through placenta. Now close to the hilum is a minute pore called micropyle. Micropyle is a minute opening in the integument of an ovule. Micropyle serves two important functions. When a seed is soaked in water, the seeds absorb water through it and make it available to the embryo for germination. These micropiles, they allow diffusion of respiratory gases for the growing embryo. Below the seed coat are two fleshy cotyledons. They contain food for the embryo. A tiny embryo is attached to the cotyledon. The embryo consists of two main parts, the radicle and the plumule. Radicle forms the root system and the plumule forms the shoot system. Now let us discuss about the structure of maize grain. Maize is called as a grain, not a seed, since its pericarp is fused with the seed coat. Maize grain consists of two unequal portions divided by a thin layer called epithelium. Maize grain is yellowish in color. The bigger portion is endosperm, which lies towards the broader side. It stores food in the form of starch, but its outermost layer contains only protein and is called aleurone layer. On the pointed end of the maize grain, lies an opaque body called the embryo. The embryo in the maize grain contains a single cotyledon called scatulum. The radicle lies towards the pointed side of the embryonic region of the seed, and the plumule is located towards the upper side of the embryonic region opposite radicle. It is enclosed in a protective sheath known as coleoptile. Some, some important questions. What is a seed? Name a monocot ex-albuminous seed. What is a micropyle and what are its functions? Describe the structure of a dicot seed. What is the difference between radicle and plumule? Why do we use the term grain and not the seed for maize? As I have told you, maize is called a grain since its pericarp is fused with the seed coat. Children, I hope you must have understood the video. Thank you for watching. Please like, share and subscribe my channel Biology with Priya Jain. Thank you.